And so it gives me great pleasure to introduce this session. It's on microbial and non-marine carbonates. And our first speaker is Nour. So over to you, Nour, if you wouldn't mind sharing your screen with us. Thank you.
Okay, assalamu alaikum. First of all, I would like to thank you to give me this chance to demonstrate my work in PhD project so far. Okay, I will talk about analyzing statistical properties and heterogeneity of Holocene freshwater from Hungary using CT data. I'm sorry.
Okay.
Okay.
Okay, start now.
Okay. So first of all, I would like to let you know a little bit about geological and geomorphological Danube to the interslave. And the Danube during the Pleistocene, before 15,000 years, it looked like spread over the Carpathian Basin. And it's changed the meandering during the Holocene. So if we look here, we can see the Danube meandering in the west part and the Tisa meandering in the east part. So the interflave covered by wind-blown sand and the Lewis and non-marine carbonate in some spot. So the hydrogeology of the area, there is many ponds cover the area related to the groundwater flows. So in the geomorphology also, we can see the sandy dune and another sandy dune. So between them, during the groundwater flow, some ponds will appear at the top. So the interflave area, and we took the cross section and it looked like this. So if we see here, so the Duna Valley is around 10 meters above sea level, and the Tisa Valley is around 80 meters above sea level. So the groundwater flows toward Tisa, and it carry magnesium ions during the journey. And the Shoyosh-Palosh, my study area, located near to the Tisa River. Freshwater carbonate rocks in the Carpathian Basin related to previous researcher. It's appear in different type of carbonate. So the dolomite here and the dolomitic limestone also spread over the area, and the lacustrine mud spread in the area too. So I focus on Shoyosh-Palosh, my study area at the 6th, and the 12th for the Shonograd, the district of my study area. The geological description, deflation valley, there is intermittent lakes. It contains soda, calcareous mud, dolomitic mud, and nitrite, KNO3. And I choose for this seminar AP sample, which describe as wind-blown sand and limonite with sandy dolomite and sandy limestone. So related to this description, and the study area looks in the wet time, fill in water, and during the dry time, it's very drought, and after evaporate, the salt precipitate over the area. So the whole cross section of my study area classified to 5 main groups, A, B, C, D, E1, E2, related to the different properties, chemical and physical properties. So we brought 4 different samples to do CT scan, and after that, try to do geostatistical analysis and define different properties. So, work procedure. We define the main content, calcite, dolomite, limonite, and while we use CT scan, so we need to know the density of these component, and combining rock types, Lewis, eoline sand, and Lucy sand, and paleosol. After that, we want to know about the process of dolomitization. In vertical layers, so the dolomite is autogenic, and sand also have dolomite grains. About the tendency in the Hounsfield unit, is it vertically increasing? And we need to know the cyclicity about the climate variation. So I plot some questions. So the first of all, why non-marine dolomite formed here? And is the climate affecting on the precipitation rates? And how statistics can define the non-marine dolomite, the climate period, and any other factors? So far, my first sample, this is AP sample, and after CT, it appears like this. So I use a subset and I try to analyze it. After got this result, I classified to this result in 5 main groups. First one related to the empty pores, and the second one related to the empty pores which filled in calcium, and the third group related to the dolomitic limestone matrix, and the fourth group related to the dolomite matrix. And in previous 10 section, we can see the calcite and the dolomite with some pores and quartz grain. And the final group related to the limonite. So if we combine all of these data, we can get the whole sample CT. Okay, for the statistical analyzing, so the histogram of this sample appears here, and it's compatible with Whitaker plot. So I try to analyze the upper outlier. It could be for limonite, and the lower outlier, it could be for the empty pores and the pores which filled with the calcite, and the dolomite limestone matrix and the dolomite matrix could be in this interval. The average block value, this is the whole block of the CT, and after that we got the slice in the Z direction. For each slice, we plot the average block value. After that, we want to know the direction under the whole blocks. We can see there is 1, 2, 3, 4, 5 different changes during the precipitation period, which related to more than 8,000 years. So it could be related to the change in the climate and chemical, physical properties of the precipitation. And this is the whole sample. We can see the different color. So it could be the limonite. There is a valuable. And I got another direction for the average block value in the CT direction to see the laterally heterogeneity. And we can see different Hounsfield values during the cross section for the different kind of material. So we can compare it with the CT of the whole sample, which increasing in the red color in this side, and in the center or almost center of the sample, decreasing of the red, and we can see the yellow and the green color, which mean decreasing of the Hounsfield unit value in the middle of this sample. For the depth average grid, to define the main component of the sample, we can see the different color, and I suppose the red and purple color related to the dolomite and limonite, and the yellow-green color related to the dolomite and limestone matrix, and the blue, light and dark blue related to the pores, and the pores which filled with the calcite. So what is the next? I want to connect the CT data with susceptibility magnetic data and to compare between a dry and wet period during the precipitation time to define the Hounsfield unit value for different types of the carbonate and to connect blocks with thin section. Thank you.
Okay, thank you very much, Nour. You kept beautifully to time. I'm just going to wait for some messages to come through in the chat, any questions. So please, the whole audience is absolutely free to type any messages here, and I will read these out to the speaker. So we have a question from All, and he says, very interesting talk and topic. Do you have a sense of the salinity within the dolomitizing environment?
Excuse me, do you mean the salinity? Yeah, it's alkaline environment, which the pH above the 11, as mentioned in the previous studies.
I think Nour is asking about the salinity. So was it, was it in any way, was it saline, very saline? Could you make do you have any constraints on the salinity?
Actually, I didn't do any test for that, but I suppose it will be high salinity.
Okay, thank you. And then we have a question from Riccardo Astini, who asks, so Nour, would these profiles plot as dolocretes in the rock record? And a second question, what can you infer of the age constraints and the preservation potential of the different outliers?
Actually, as I mentioned here, so my sample related to the previous studies, it's ages between 15,000 years and 12,000 years. So all the components related to this period.
Okay, thank you very much. And then we have a question now from Fiona Whitaker. Is there any sign of alteration of these sediments after deposition?
It could be because the magnesium come after the precipitation of the calcium carbonate. So maybe, so the dolomite, it could be not autogenetic, after the autogenetic of the carbonate. So it added the magnesium ion and product or form protodolomite in this area.
Okay, thank you. And then we have the questions are pouring in now. What type of dolomitization is present? Is it reflux, or do you think it's another type?
It could be reflux because it's the main source of the water is the groundwater, and it contains the ions during the travel from the Danube to the river. It could be.
Okay. Yes, thank you. We've got two more. Oh, they're really pouring in. Sorry, one moment. I just need to. They're pouring in. I have to come back up again. Do you have any suggestion of any microbially mediated precipitations for any of these phases?
It could be for the next step, actually. So the first step I did just to get the whole idea about the project and try to analyze the whole CT scan data. And the next step, it could be more specified and focused more in the subject.
Yes, that would be very interesting. And the next question is, have you quantified any of the various phases that you've identified?
Not yet, not to quantify as in the meaning, but just to get the whole distribution and analyze the whole sample. The next step I will do the get the numerical data for each slice and analyze the slice by slice for get more information about the study area.
Thank you. And then we have another question. Sorry if this is from Omar. Sorry if he's missed it, but what is the statistical method which you've used to define the non-marine carbonate?
Actually, it's simply a statistical analysis because we got the numerical data from the CT, and after that, we try to get the tendency and to define the mean and get the some numerical analysis from different software and collecting this data and try to analyze it as simply procedure.
Okay, thank you. We have time for just a few more questions, a couple of minutes. So a question here from Fiona Whitaker. Do you see any hydromagnesite?
Not yet, but maybe when I do science section, the next step, it will be defined if it's valuable or not.
Thank you. So Fiona says thank you. So thank you very much for those questions. We may have time just for one more before the next talk. So if anyone wants to quickly type one in, I can read it out. Yes, Fiona, where does the limonite come from? She says, sorry, lots of questions from her, but she's really curious.
Okay, thank you. And I hope I can answer it. I suppose because the groundwater will move from Danube to Tisa and it's cover a huge area. And there is sandy dune come from different places. So it could be bring the different kind of mineral that may be the reason to available the ions in the water, and after that bring different kind of mineral.
Well, thank you very much, Nour. Thank you for sharing your study. It's obviously provoked a lot of questions, so thank you very much indeed.
Thank you for you.
So thank you. So if anyone we have no more questions, I think we'll move on now to the next out rooms at the end of the session when we can really get our teeth into these topics. So we have our next speaker now, which is Tom. Who is Tom? Tom, I think you're about to come online.
Yeah.
Lovely, thanks, Tom. So Tom is going to talk about microbial versus abiotic carbonate precipitation and diagenesis. So over to you, Tom.
Thanks, Rachel. Hi, I'm Tom Kibblewhite. I'm a PhD student at the University of Bristol. Today I'll be presenting the results of some field work looking into the controls on carbonate precipitation and diagenesis in a hydromagnesite playa in British Columbia. Everyone here should have heard of calcite and magnesite and dolomite, but you might not know what hydromagnesite is. The hydromagnesite is a metastable hydrated magnesium carbonate associated with the alteration of ultramafic rocks by hydrothermal or groundwater fluids. Hydrated refers to the water in its structure, magnesium to the magnesium, and carbonate to the carbonate. But metastable is referring to the fact that hydromagnesite eventually dehydrate, losing its water molecules to form magnesite through a series of other intermediate magnesium carbonates. So why do we want to study hydromagnesite? We'll first look at saline lakes, which form in closed basins dominated by evaporation. They're important sources of evaporitic salts and associated elements. Soda lakes are a subset of carbonate-rich saline lakes that present a potential analogue for the South Atlantic pre-salt that Nathan and JP will be covering in their talks next. Now, hydromagnesite playas are soda lakes, but they have an additional interest of being relatively rare and potential analogues for CO2 sequestration. The rest of this talk will focus on the results of fieldwork on a hydromagnesite playa in British Columbia. Atlin town and its playas occupy a small endorheic basin on the east coast of Atlin Lake. Like most basins, it's very arid, lying in the rain shadow of the Rockies and Juneau icefield. But unlike most endorheic basins, it sits on the Cache Creek terrain, which is an ultramafic ophiolite complex of altered oceanic crust, mantle minerals, and limestones, from which the groundwaters dissolve the ions required to precipitate the carbonates. So there are 3 hydromagnesite playas in Atlin, and we'll be looking at one of those today. The surface is composed of a complex mosaic of different environments, including several wetlands and microbial mats within them, areas of grassland, and actively precipitating hydromagnesite mounds of varying morphologies. If we look at this in cross-section, we can see how these evolve. So the groundwater-fed wetland becomes saline as its waters have evaporated, and calcium carbonates begin to precipitate. As these carbonates fill the wetland, they emerge from the water table and are colonized by grasses. Precipitation then continues in the emerged sediments to form localized mounds of what is now dominantly magnesium carbonate. Over time, these incipient mounds that coalesce to form larger amalgamated mounds of magnesium carbonate. In order to assess the processes of carbonate precipitation across the playa, we sample pool waters and sediments at the top of the mound and at the edge of the wetland. To collect samples, we dug shallow boreholes in the playa, and pore waters were extracted under vacuum through ceramic rhizon filters that are inserted at various depths to create a profile. Pore waters were then analysed for pH, EH, and alkalinity in the field and sent back to Bristol for ion analyses. Rhizons were then removed and the syringes used to take sediment cores over the same profile. Some of these sediments were sent to the University of Oklahoma for DNA sequencing, whilst the rest was dried in as awaiting mineralogical analyses. In addition to pore waters, we also sampled the playa's input waters and surface wetlands. You can see that bicarbonate and magnesium are the major ions in all the waters, and their concentration is lowest in the groundwater and highest in the pore waters. Alongside this, calcium is relatively low in most of the waters, producing incredibly high magnesium-calcium ratios of up to 10,000. For comparison, the Mg-Ca ratio for seawater is only 5. And if we look at the pore waters in more detail, so we have the wetland site, which has a water table at 2 centimeters, so most of the sediments are water saturated. Above the water table, there is a thin crystalline carbonate crust with red and green staining, and below the water table, the sediments are poorly consolidated and transition through different organic rich brown and grey layers. Over the profile's depth, there is a change in 1.3 pH units, and all of the waters are reducing. At the same time, there is a clear redox zonation with peaks in nitrate, sulfate, and methane. This is common in most water-saturated sediments and caused by the preferential use of more efficient metabolites by microbes. Conversely, at the top of the mound, there are these cauliflower-like desiccation structures and polygonal fractures extending deep into the mound. All the mound sediments are bright white, apart from some crystalline hard pans in and around the water table that are slightly yellowed. Prior analysis shows that all of these sediments are hydromagnesite and magnesite. The range of pH in the mound is much smaller than the wetland, and all of the waters are oxidizing as opposed to reducing. And there is no semblance of a redox zonation, suggesting a relative absence of microbes in the mound. We can look into this with the 16 RNA analyses. You can see that the microbial community in the wetland is much more diverse, with 5 different phyla with a fairly high abundance, whereas only proteobacteria and actinobacteria dominate the community in the mound, as well as some component of dependentia which are not at all present in the wetland. Principal coordinate analysis of the same samples identifies a change in both communities with depth, and also shows very clear difference between oxic communities and anoxic communities. And now I'll relate all this to carbonate precipitation with mineral saturation indices. A negative saturation index suggests that mineral is likely to dissolve from solution, whilst a positive saturation index suggests that a mineral is likely to precipitate from solution. So we have hydromagnesite. In both sites at the surface, it's fairly supersaturated, and for the whole depth of the mound site, it remains supersaturated, whilst in the wetland site, it becomes undersaturated with depth, indicating possible dissolution. You can see that in both cases, this correlates strongly with pH, which is more variable in the wetland site, hence the more variable saturation index. We also have aragonite saturation indices, which throughout the wetland and beneath the water table in the mound are near equilibrium and slightly supersaturated, whereas above the water table in the mound, they become undersaturated. This time it anti-correlates with the Mg-Ca ratio that we mentioned earlier. We can draw a line between the phreatic sediments below the water table and the vadose samples at about 850 Mg-Ca ratio. And I'd suggest that this is a potential threshold for defining the difference between calcium carbonate precipitation in the wetland and magnesium carbonate precipitation in the mound. So to conclude, redox in the wetland is very strong and redox in the mound is near absent, which correlates with a diverse microbial community in the wetland and a near absent microbial community in the mound. Mg-Ca ratio in the wetland is less than 850 and also less than 850 in the water saturated zone of the mound and greater than 850 in the vadose zone of the mound. This correlates with the precipitation of calcium-rich carbonates in the wetland and magnesium-rich carbonates in the mound. And that's all. sorry, I don't have an acknowledgement slide, but I'd like to thank Hamish Robertson and Fiona for the help in field work, and our colleagues in Oklahoma for their samples, and other colleagues in Bristol for their help with lab work and analyses. Thank you.
Thank you very much indeed, Tom. The chat's now open for anyone to send in questions. And just a reminder, please do send them to everyone rather than privately. I'll just ask you a question, Tom, while we're waiting. So a wonderful integrated study. I don't know whether this was a subtext, but the inference or the recording of a very limited, a non-diverse microbial population associated with the mounds.
Mm-hmm.
I'm therefore suggesting that those mounds have very little microbially mediated precipitation of carbonate, or that the carbonate is dominated by the precipitating powers of a very limited number of microbes.
Yeah, I think that there's almost certainly mediation of the precipitation by microbes, where, as I say, we're not exactly sure what's doing it, but they're clearly there. Whether they're reactive, we don't know, just from looking at the RNA data. We don't know whether they're alive or dead, but there are definitely microbes there. And the dependency that only appear in the mound and not in the wetland is interesting. Perhaps they're quite important, but it's complicated and we're not really sure. Apart from things like cyanobacteria, we're not really sure what they do.
Yes, it'd be interesting to know what role they're playing. And in fact, that was one of the first questions that has come in, how important, how important was the control of the microbial population on the type of carbonate precipitated?
So yeah, we're not sure exactly, but we know that there's a clear difference in community and that microbes are present everywhere and provide ample surface for mediating precipitation, whether that's intentionally or not. But no, we're not sure exactly what the correlation is.
Yes, thanks, Tom. I should have said that was a question from Madeline Vickers. And now we have a question from Hal Bradbury. Interesting talk. Why is it that the redox zonation causes very little change in pH in the wetland sediment?
I think it does cause quite a bit of change in pH. I mean, pH changes over 1.3 pH change over just 30 centimeters of depth. So even though the zonation only occurs over the top 10 centimeters, there's still sort of 0.7 pH drop, which I think is fairly significant.
Yes, thank you. And then we have a question we have loads of questions pouring in, Tom. A question from Maya. Do you have an idea about the cell density or numbers of microbes within the sediments?
So Powers, who I referred to earlier, he may have had some cell density data, but no one's done profiles like this, and we were unable to do that at the time. So no, we don't have any idea really, apart from looking at other similar pliers, but no, not from this site.
Okay, thanks. I'm going to storm through these questions because there are lots and some of them are very long. So Madeline Vickers asks, was there any evidence of formation of biofilms?
Biofilms. We couldn't see, there's no fabrics evident within the sediments, but there are these thick gelatinous brown mats within the wetlands and isolated pools that you have, again, a sequence of colouring of purple, green and red layers within these microbial mats. And presumably there are biofilms, but we've not looked at anything under SEM or anything to determine any more than that.
Thanks. And then Chelsea asks, hi Tom, interesting study and great presentation. 2 questions. One, does the water table fluctuate much annually? And could this help explain the difference in the chemical variability between the environments? And the second part, I'll just quickly, and the second part, I'm surprised by the high supersaturation in the wetland for hydromagnesite compared to aragonite, yet modern precipitation is preliminary aragonite. Did I get that right?
So the first one about water table. So the water table does fluctuate quite a bit. There is, in the summer months, it's much more evaporation than in the winter months. And compared to the sampling we've done and previous sampling on the playa, the water table has been sort of 40 centimetres difference. And over the course of being there for several weeks, you could see the water table had dropped by several centimetres just in that time. And I think that if we look at mineralogical data from previous studies, I've not put it in here, but there is not a direct correlation between the actual saturation index and the percentage of calcium or magnesium minerals, rather a broad, if their ratio is above this 8 or below this 850 sort of threshold, there is a dominance of calcium or magnesium minerals. And I think the reason that's blurred is because the water table is moving around and therefore the chemistry of the pore waters is fluctuating a lot. And there's also diagenetic factors given that these are metastable minerals. And then the hydromagnesite. So yeah, hydromagnesite is still, like Chelsea says, is supersaturated compared to the aragonite in the wetland. There is some magnesium carbonate precipitation in the wetland, just not as much as in the mound. And yeah, I'm not sure exactly why there's not more, but we think it's to do with the Mg ratio, just that it's not happening fast enough at the lower ratio.
There's loads of questions pouring in. I'll try and get through as many of them as I can, but I think this is obviously something for a really good breakout session later. Just another question from Raphael Peach. Nice study. What is, again, the difference between the dashed lines and the full lines of the figure that you have up now on the screen, just to quickly tell the audience?
Yeah, I'd go for that. So the shorter lines down to 33 centimetres that are dashed are the wetland site, and the deeper ones down to 82 centimetres of the mound site. Yeah, it doesn't come across very well in the figure, but there. Yeah, dashed is wetland, solid is mound.
Thanks, Tom. So there are a lot of questions here. Martin asks, do your results mean that the microbial respiration does not play a role in precipitation, but only in the saturation index?
So saturation index is sort of a proxy for precipitation, and it states whether it's thermodynamically likely for things to precipitate, but there's also a kinetic factor to precipitation. For example, I've not plotted magnesite and dolomite on this graph, but they sort of have saturation indices of 3 and 4, and so you'd expect them to precipitate when actually kinetically you have to overcome this energy barrier for them to precipitate. And so, yeah, they may, that part of it, I'm not sure if that's the question. But.
No, that's great. There are many questions here that I don't have time to read out because we've got to the end of our time, and some of them are long. Obviously, a lot of interest, Tom, in your talk, so great stuff, and obviously something for a breakout room later. We'll capture these messages and we'll put you together with those that wish to talk to you afterwards. So many thanks, Tom. Hi there. Modern lacustrine carbonates. Over to you, Connor.
Thank you very much. So today I'm going to talk a little bit about some of the work we've been doing on modern and ancient microbial Mg and Ca-carbonates of the Iberian Peninsula. So I'll give you a bit of intro to the background of this study, some of the problems and the outstanding issues, and some of the aims and objectives we've been taking, and the overall approach that we have on this work. And I've more or less split it into the modern and Holocene, sort of older microbialites we've been working on. So, to move this over. So microbial mats are found in lots of different modern and ancient environments, and obviously when they become rocks, we term them microbialites. In these mats, the microbes can interact with carbonates through the metabolic activities. So for example, if we consider carbonate precipitation, for example, processes of photosynthesis and sulfate reduction can change, for example, the concentration of calcium Ca2 plus ions, or they can change the pH. This can lead to processes of precipitation, or in some cases dissolution, for example, in the process of sulfide oxidation. So in this sort of field, there's a couple of key questions and that I'm going to sort of talk about here and what importance they have. So one is, are there actually some diagnostic structures, minerals and textures we can look at, and can we associate those with specific geochemical conditions? So obviously that's quite important for, I guess, categorizing how geochemistry and mineralogy interact together. How do these microbial carbonates actually evolve when it comes to diagenesis? And how do these modern microbial mats become stromatolites that we find in the geological record? So obviously that's quite important for understanding diagenesis as a whole, but also for means of analog modeling. And then we want to know as well, how do spatial and temporal variations in geochemistry within different settings actually affect the morphology of these mats and their mineralogy? So it's quite important for understanding the biogeochemistry of these deposits, and it's obviously very useful for understanding early earth processes. So the aim of this study has essentially been to look at spatial and temporal variations in the crust in geochemistry and see what effects this has upon microbe-mineral interactions and diagenesis in these mats. So to do this, kind of three objectives we've got. So we look at microbe-carbonate interactions in modern and ancient Lacustre environments using XRD, XRF, microscopy and CT. We want to look at mechanisms of early diagenesis using obviously standard sort of petrographical comparisons between modern and ancient examples, textural classifications, and we want to look very sort of, as on a more basic level, what are the actual biological communities that are leading to potentially leading to carbonate precipitation and dissolution, and how does that balance in with abiotic processes? So we can study microbialites of varying age and in different geochemical settings in the Iberian Peninsula. So there's a wide variety of Lacustrine settings here with microbialites of different age. Basically, when I split this up is we have these contemporary microbial mats, so between 1 and 20 years old. We have these three settings here, hypersaline, saline, and alkaline. So we find these types of mats on the right here in these settings. We have older microbial mats, which are sort of between 1 and 10,000 years old, at these three sites I've just highlighted here. So we've got a good archive of the environmental conditions that have occurred during mat formation in these systems. So we know roughly what the conditions of formation were at the time. And then the final sort of part of the study is to look at much older stromatolites in the Ebro Basin in northeast Spain. And these are about 6 million years old that were forming in a hypersaline playa lake sort of setting. So I'm just going to go into some details on some of the work we've been doing on Laguna Salada Chiprana, which is a hypersaline lagoon in northeast Spain. So one of the main things we've been doing is actually growing microbial mats on the Solnhofen limestone plates. So we placed these into the lake I think March last year. And after 8 months, this is what we see. Quite a nice growth of this top gypsum crust here, this pinnacle gypsum crust. Green sulfur bacteria below that, and then this black layer at the base, which is characterized predominantly by sulfate-reducing bacteria. And we can overlay there just to kind of get a better idea of what things we can see that are developing on these plates. If we look at that in more detail, we can actually see quite a common characteristic is this kind of very interconnected sort of network we see here, and this pinnacle formation. And this is most likely driven by environmental gradients in salinity, in light, and competition by microbes to essentially survive within the mat. And we've been doing this across the entire sort of lake, essentially. So this is a very sort of basic cross-section showing some short sediment cores we retrieved from the lake. And these essentially have been scanned and photographed, and we're looking at the different morphologies of mats at different depths in the lake and comparing those and seeing essentially what controls are acting upon them. So when we put them under the microscope, we can see that there's these main sort of three textures we can see here. There's like a clotted texture, there's the pinnacle texture, and there's a very strongly laminated texture. And generally, it's a mixture of siliciclastics, carbonates, and organics here. Generally, the microbial horizons are closely associated with high concentrations of carbonates and magnesium, calcium. And as I say, these are the main kind of textures we see just within one lake. So there's quite a variety of processes leading to the formation of these. From a more geochemical perspective, they're dominated by carbonates and evaporates, obviously. We have gypsum as the predominant evaporitic phase. Hexahydrite is a magnesium sulfate, as this lake has quite a high concentration of Mg2+. And then aragonite tends to be the predominant carbonate mineral alongside calcite. When we try and look at where these actual phases are within the mats, this is actually a cut down the middle of a mat we sampled last year in November, and it's been put under the SEM using EDS. We zoom in, we can actually see that pinnacle we saw before actually has quite a high concentration of magnesium. This is obviously very useful for seeing where the carbonate is versus, say, for example, the calcium sulfate, the gypsum in the samples. So this is very representative of where we were looking for this carbonate in the future using high magnification SEM to see what the microcrystalline phases look like. And just in more detail in geochemistry, I'm not going to go into too much detail on this, but we've done very high resolution XRF analyses of these cores, basically because we want to get as much background information about the processes that were occurring during periods of microbial mat formation. You want to best constrain what processes were leading to mat formation and what the geochemical conditions were at the time. So this has obviously been done with XRF, which is obviously qualitative, but it's very useful for understanding different paleolimnological processes. And then we've also done this with XRD. We've produced the down-core XRD records, facies analysis, and total organic carbon. And this is just, as I say, to get a much better idea and background of what processes are occurring, not just in the modern day in this lake, but also in the past. So kind of moving on to some older mats from other lakes we're looking at. These are from a site called Laguna Toñar, which is in southwest Spain in Cordoba. So these are about 500 years old, and these have been CT scanned. So you can see here very nicely the actual morphology of the mats. It's very easy to determine where different compositional phases are and where we want to target high magnification SEM analyses. And if we can actually animate these, so this is one example that has been animated, and you can see very nicely how the morphology of the mat changes across a 5 centimeter transect there. So this is very useful for understanding how the morphology changes, but also where different phases are concentrated within the mats themselves. Under a microscope, we get some very nice textures forming in these types of mats in some cases. There's a very nice stromatolitic domed feature here that has an alternation of different phases in the actual mat itself. So you can see on the right there, under cross polars, we have probably an alternation of organic rich horizons with carbonates, siliciclastic horizons that may have been trapped and bound by the mat itself. And then, of course, there's bits of porosity that are scattered about in this section. We want to go a little bit older. This is in Lago de Estaña in northeast Spain, just in the Pre-Pyrenees. And this is actually very similar to the textures we just saw in the previous presentation, this kind of lamella interconnected fibrous network we see. And we see in here diatoms are present as one of the microbial communities. I think in the center there, it's difficult to tell, but we potentially have some small algae in the center. But as I say, so we're seeing very different textures in different lakes and in different ages of mats. So it's interesting to see, and it'd be interesting to find out what's actually causing these changes. When we look at this under the SEM again, we can begin to understand where the carbonate is versus the gypsum, calcium sulfate. And on the left there, we can see the calcium is where the blue is concentrated. On the right, it's where the green. So this has been done prominently to see where the carbonate is and where we want to focus those high magnification and SEM analyses.
2 minutes to go, Connor.
Thank you. So yeah, as I say, it's very easy to see where the different phases are, and this is kind of forming the foundation for what we are doing in the future with this study. I'll just start to wrap up now. So there's a wide variety of textures and fabrics between the different materials and different sites. The textures in mineralogy essentially, I think, are key to beginning to understand what geochemical conditions were occurring, particularly in the past, and we can do that by modelling the processes in the modern. We also need to consider that within lake variations in geochemistry and external forcing factors such as anthropogenic processes, climatic processes, how they contribute to textural and mineralogical variations. And the CT scan is very useful for looking at potentially in much greater detail what biological and environmental controls were happening in these mats. So in the future, we're going to carry on visiting Spain and looking at the microbial morphology and mineralogy, begin to highlight in more detail what processes are happening, the biology of these mats and precipitates, begin to see what processes of early diagenesis are happening. I'll just leave you with some of these pictures and these acknowledgements. Thank you for listening. I'll be happy to take any questions.
Thank you very much, Connor. We've got some questions coming in. First, from Stephen Lockyer, great talk and some beautiful images. Sorry if I missed this, but how are you dating the facts?
So in Ciprana, we have a cesium record, cesium-137, which we're linking to post-World War II radionuclide tests, and we also have a lead-210 record. In some of the other lakes, carbon-14 specific data have been used as kind of, horizons of, say, for example, 2,000, 3,000 years ago. It's difficult to tell, obviously, the exact age of a mat, but these are useful at least to see roughly what period they were developing in. But as I say, in the lakes where there's been microbial mats identified in the Holocene record, those lakes have been studied in detail before. It's just we're looking in much more detail at the mats themselves in the succession. Okay,
thank you. And a question from Bembio. Excellent work. What is the lateral continuity of the gypsum interval from vertically? Is it from vertically millimeters to centimeter scale?
Gypsum interval. Sorry, could you just elaborate on that? Sorry.
I think it's just what is the lateral continuity of the gypsum interval? I think that's what's being asked.
Gypsum interval. I'm just trying to figure which interval he's talking about.
Benbia, perhaps you could write another little question to elaborate there, if you don't mind. Perhaps I could just ask a question.
Yeah, sure.
That's coming online. I noticed it was a fleeting image, but some of the scans you had showed very discrete layers that were rich in magnesium.
Mm-hmm. Sorry.
I can't remember which slide.
This one here. This one here.
Sorry, I've got the chat going here. Yes, exactly. Yep. Can you say a little bit more about that?
Yeah. So in this case, as far as I know, if you look at the overall XRD, so this is actually from the same sample, I believe, but I'm attributing the Mg to be coming from the aragonite and potentially high Mg calcite. It's interesting that we don't actually see any calcium at the top there, as you can see in that bottom left scan. So in this case, I'm assuming you're just getting a lot of this magnesium concentrating in the mats, whether that's it's most likely by photosynthesis processes such as that, which are concentrating this carbonate either on the bacterial cells within the EPS, the extracellular polymeric substance. But as I say, it's quite interesting to see, well, to figure out why there actually isn't any calcium in that interval, but such a strong, concentration of that magnesium.
Okay, thank you. Ben B has just followed up. He's asking, is there any relationship between the vertical thickness and the lateral continuity of the gypsum intervals?
I don't think it's something I've actually thought about, to be honest. It's not something, at least, we're looking at. I mean, the gypsum intervals especially in Ciprana, are very sort of interesting in the sense of in the sort of more shallow settings in this lake, you actually get this really sort of distinctive pinnacle almost like a cumulus gypsum texture, whereas in the much sort of deeper setting, so maybe like, well, not much deeper, but only like a couple of meters, but basically where there's probably been a bit of a change in the halocline and the chemocline of this lake, and where there's much less light intensity, you're getting actually a different sort of development of gypsum. It's much more dendritic, and it seems to displace the actual microbial mats that are developing down in the deeper parts of the lake. So whether there's actually a microbial component affecting gypsum growth, and if that's affecting how gypsum is developing in different parts of the lake, as well as the carbonate, that's something we're definitely looking at in this this lake. But as I say, in terms of the lateral continuity and the thickness with that relationship, it's not something I've actually thought about, but I hope that gives a little bit of clarification at least.
Okay, thank you. And we have a couple more questions. One from Fiona, two from Fiona actually, but I'll go for the first one first. How important do you think the differences in lithology of the basin hosting the lakes might be? In other words, differences in
I think absolutely. It's definitely important, because just in this lake alone, if I just go to this slide here, if you look at this interval actually down here, you can see you get a massive increase of what would predominantly be considered chiral elements, silica, potassium, titanium, iron, and actually we get a bit of an increase in calcium there as well. But we also get an increase of calcium here. Now that increase there can actually be attributed to the fact that in Ciprana the lithology is actually quite marl rich. So we do get for example, in this period, we've kind of looked and said it's most likely we've had some more detrital input here, which has brought in actually a lot of detrital calcium. But then, as I say, it's separating that detrital signal from the endogenic signal, for example, and that becomes a bit of a challenge in these lakes. But it's something that can be done. I guess you can use, for example, the strontium ratio, which gives you a better idea of in situ carbonate precipitation, as you can see here. But I think absolutely it is important, and just in these mats you do see different sort of trapped material in the mats, and different phases that you can see between different sites. So yeah, definitely, it's definitely important.
Okay, thank you. And Ricardo asks, nice talk and work in progress, Connor. What about the gypsum crusts? What is left after an early diagenetic imprint? And is there any deformation?
There doesn't seem to be, any deformation. I think maybe the only potential diagenetic thing you may be getting, I doubt it, but potentially some anhydrite formation. Probably not, considering it's only very, very shallow depths. But one mineral that I have noticed a little bit occurs in these sediments is thenodite, and it's this little mineral we have here, which as far as I'm concerned is actually an anhydrous evaporitic mineral. So whether there's some relationship there between gypsum and thenodite, whether there's actually whether the diagenesis is coming from physical or biotic processes is I'm unsure about. But the gypsum itself, I can't say there's a massive amount of diagenesis occurring just in 50 centimeters at least. Maybe below this you're getting potentially a little bit of anhydrite formation from gypsum, but as I say, in this section we haven't seen it from any of the XRD scans, XRD diffractograms.
Thank you. And just one very, very brief question that I think just needs a very brief answer. Fiona just wants to ask a follow-on question. Mm-hmm. Do you think these lakes are deep enough to have a chemocline?
I think in the case of Ciprana, I can't say for definite at the moment. I know in the past they've done a study, and there's definitely a halocline. We were planning to be, well, I think we would have actually been in Ciprana right now only for this, but we were going to take a limnological profile. I know in Lago d'Estagna at the moment that has a little bit of a chemocline, but that's 29 meters deep, I think. Maybe I think in Ciprana it's probably not as pronounced, but as I say, we don't have a limnological profile of that lake at the moment, but it's something that once we get back out in the field, we can actually clarify that and see if there's any, major difference between the sort of 1 meter depth versus 4 meter depth, and if that's affecting microbial processes.
Fiona says, thank you very much. And I'm going to unfortunately have to stop the questions. There are all coming in, but we've slightly run out of time. But thank you very much indeed, Connor, and thank you to all the speakers. What a vibrant session, really great. So many, many thanks. And I'm going to hand over now to Kathy, who'd like to make some announcements. Thank you.